DNA modification. DNA modification is the process of changing the genetic material of an organism by adding, deleting, or altering specific genes. This technology has become an important tool in numerous fields, including biotechnology, medicine, and agriculture. DNA modification techniques have led to the development of genetically modified organisms, GMOs, which are organisms that have had their DNA altered to improve specific traits or characteristics. One of the most common applications of DNA modification is in agriculture. Farmers can use genetically modified crops to increase yields, reduce the use of pesticides, and improve resistance to environmental stresses, such as drought or extreme temperatures. In medicine, DNA modification techniques have been used to develop gene therapies to treat genetic disorders. Gene therapies involve introducing a functional copy of a gene into the patient's cells to replace the defective or missing gene. There are several techniques used for DNA modification. One of the most widely used techniques is recombinant DNA technology. This technique involves cutting DNA at specific locations and recombining it with DNA from other sources to create a new DNA molecule. Other techniques include RNA interference, RNA, which can be used to selectively silence specific genes, and genome editing, which uses enzymes to cut DNA at specific locations and insert, delete, or modify genes. Despite the numerous benefits of DNA modification, there are also concerns about its safety and ethical implications. Some critics argue that genetically modified organisms may have unintended effects on the environment or human health. Others raise ethical concerns about the use of DNA modification techniques in humans or animals. Overall, DNA modification has become a valuable tool in various fields of science and technology. With ongoing research and development, it has the potential to continue improving agriculture, medicine, and other areas of science. However, it is essential to consider the potential risks and ethical concerns associated with this technology and to proceed with caution. Molecular biology. Molecular biology is a field of biology that focuses on studying the molecular basis of life. It is concerned with the study of DNA, RNA, proteins, and other molecules that make up living organisms. The field has grown rapidly in recent years due to advances in technology and has made significant contributions to our understanding of genetics, biotechnology, and medicine. One of the main areas of research in molecular biology is genetics. The study of genetics involves analyzing the structure and function of genes, which are the basic units of heredity. Researchers use molecular biology techniques to identify specific genes and understand how they are regulated, expressed, and mutated. This knowledge has led to the development of genetic engineering and genome editing techniques, which can be used to modify and manipulate genes to improve human health and agriculture. Another important area of research in molecular biology is biotechnology. Biotechnology involves the use of living organisms, cells, and molecules to produce useful products and processes. Molecular biology techniques are used in biotechnology to modify and engineer living organisms, such as bacteria and plants, to produce specific products or to perform certain tasks. For example, molecular biologists can modify the DNA of bacteria to produce insulin, which is used to treat diabetes. Molecular biology has also made significant contributions to medicine. The study of molecular biology has helped researchers understand the genetic basis of many diseases, including cancer, Alzheimer's disease, and heart disease. Gene therapy. Gene therapy is a promising field in biotechnology that aims to treat genetic disorders by modifying or replacing a patient's defective genes. The goal of gene therapy is to introduce healthy genes into the body to replace faulty ones and restore normal function. The process of gene therapy involves identifying the specific genetic mutation that is causing the disease and designing a therapeutic strategy to correct it. There are two main approaches to gene therapy: ex vivo and in vivo. Ex vivo gene therapy involves removing cells from a patient, genetically modifying them in the laboratory, and then reinserting them back into the patient's body. In vivo gene therapy involves delivering therapeutic genes directly into the patient's body using a viral vector or another delivery method. Gene therapy has shown promise in treating a variety of genetic disorders, including inherited immune deficiencies, hemophilia, and some types of cancer. It has also been used to treat rare genetic disorders, such as spinal muscular atrophy and cystic fibrosis. Gene therapy has the potential to provide a cure for these diseases, rather than just managing their symptoms. Despite its potential benefits, gene therapy is still in the early stages of development, and there are many challenges to overcome. One of the main challenges is the development of safe and effective delivery methods for therapeutic genes. Researchers are also working to better understand the long-term effects of gene therapy and to develop strategies to minimize any potential risks. In conclusion, gene therapy holds great promise as a treatment for genetic disorders. As research continues to advance, we can hope to see more successful gene therapy treatments for a wider range of diseases.